All right, this video is going to cover Chapter 3, Prokaryotic Cell Morphology, Structure, and Function. This is one of the more important chapters, not only for the first unit, but also for the entire semester. You will need to know the bacterial cell structures for the entire semester. This first image shows just a typical prokaryotic cell. Typical prokaryotic cell, this would be a bacteria, is around 1 to 5 micrometers in length or diameter, depending on the type of bacteria. We'll cover each of these highlighted items as we go. Beginning with the cell membrane or cytoplasmic membrane, the structure is composed of two layers or leaflets of phospholipids. There is a hydrophilic head and there is a hydrophobic tail. So the heads are phosphate groups and the tails are the lipids. There's various proteins inserted throughout the phospholipid bilayer, which would be involved in transporting nutrients and waste. The structure is defined as a fluid mosaic model, which basically means that the proteins and the phospholipids in the cytoplasm or the cytoplasmic membrane are not fixed in one location, but they are fluid and they do move about. Here we can see an image with the hydrophilic, water-loving heads and the hydrophobic tails oriented inside the phosphate groups. As you can see here, there's the different proteins located throughout the membrane. The function of the cell membrane, like I mentioned, is for transporting nutrients, but also the site of energy generation if they have an electron transport system. Not all bacteria have an electron transport system. We'll cover that in detail in Chapter 8 when we do cover metabolism. But it's a permeability barrier which contains the cell contents, so that contains the ribosomes and the DNA, for example. It's selectively permeable. Only a few types of molecules can actually pass through the membrane freely. And as I mentioned, again, transporting nutrients, typically against a concentration gradient, which requires energy. Looking inside the cell, we have the cytoplasm, which is the liquid portion of the cell itself. And this is where most of the biochemical and functions occur inside the cell. The DNA is located in a concentrated area referred to as the nucleoid region. Over here on the right-hand side, this red concentration of the DNA is highlighted here in these two cells. So the chromosome is singular and circular, indicating that they only have one chromosome, unlike other types of organisms. And some cells actually have what are called plasmids, which are referred to as extrachromosomal elements, meaning they're not part of the actual chromosome itself. And these plasmids have genes, so sequences of DNA, that are not essential for normal cell function. So they often carry genes for antibiotic resistance. Ribosomes are part of these, found inside the cytoplasm, and they serve as the site of protein synthesis. Each ribosome is composed of a large subunit and a small subunit, which is indicated by an S. And the S basically indicates the density of the large and small subunits. The prokaryotic ribosomes are referred to as 70S ribosomes, which is composed of a 30S small subunit and a 50S large subunit. They do not add up to 70, as indicated by the 70S, but this, like I said, the S is basically the density of the large and small subunits. Eukaryotic ribosomes, so ribosomes that are found inside our cells, are actually 80S, so they are different than prokaryotic ribosomes. Eukaryotic ribosomes are composed of a 40S small subunit and a 60S large subunit. So that's one of the main differences between a prokaryotic and eukaryotic cells uh, other than the lack of a nucleus. So one of the interesting things about eukaryotic and prokaryotic ribosomes is that inside eukaryotic cells, our cells, mitochondria actually have 70S ribosomes. So there's this theory called the endosymbiotic theory, or endosymbiont theory, which basically states that ancestors of mitochondria and chloroplasts were once free-living bacteria, and they basically formed a beneficial partnership. The mitochondria, if you remember from other classes, is the powerhouse of eukaryotic cells, which is the site of the electron transport system. Chloroplasts are found in plant cells, and those are the sites of photosynthesis. So some evidence of this endosymbiotic theory are that the mitochondria have genes, like I said, that make 70S ribosomes. Other parts of or outside of the mitochondria, eukaryotic cells have 80S. DNA comparisons show that of the mitochondrial DNA have close relationships to a group of obligate intracellular bacteria. And they also elongate and divide similar to bacteria, and they have a double membrane. I posted a link in the e-learning website, so you can click on this YouTube video to further explore the endosymbiotic theory, kind of outlining some of this evidence. Other internal structures in some bacteria, I want to highlight some bacteria, not all bacteria have these inclusions. They're also called granules. These granules are basically large vesicles that store certain molecules, so starch, glycogen, sulfur, and even magnetic particles. So this bacteria down here on the bottom right has these magnetic particles, allowing them to fix to certain metals in the environment. They can also contain gas, which would allow them to be buoyant in a water system. This image here shows in large granules of this from this bacteria. It's not very important, the name of it, but the molecules that are found in here, it's called polyhydroxybutyrate, and this chemical is used to make biodegradable plastics. So you can grow these bacteria in large quantity, harvest this polyhydroxybutyrate, and use that to create biodegradable plastics. One of the more important topics in this chapter is regarding the bacterial cell walls. We can divide bacteria into two large groups of organisms referred to as either gram-positive or gram-negative bacteria. Gram-positive bacteria have cell walls contained of peptidoglycan. So peptidoglycan is a term that you'll need to know what it is and differentiate between gram-positive and gram-negative organisms. So gram-positive have thick layers of peptidoglycan. We'll talk about the structure of peptidoglycan here in a minute. Gram-positive bacteria also have teichoic acids that aid in the passage of ions, and lipoteichoic acids that anchor the cell wall to the cell membrane. So the cell wall is located outside of the cell membrane. One thing about gram-positives is that they appear purple following what's referred to as the gram staining procedure. Some gram-positive organisms, such as organisms from the genus Mycobacterium, can contain up to 60% mycolic acid in what are referred to as acid-fast bacteria. This helps the bacteria survive desiccation or dry environments. So here's an image of peptidoglycan showing you alternating subunits of what's referred to as NAM and NAG. So here in blue is NAM, NAG in purple. And you can see they alternate NAM, NAG, NAM, NAG. So this is one layer of peptidoglycan. Here's another layer of peptidoglycan, another, and another. So there's 4 layers of peptidoglycan in this image. And here they are also cross-linked to each other. And what we like to refer to this as kind of a chain mail armor, something like you might find in a medieval movie. And what the cell wall does, and the peptidoglycan does, is it protects the cell from osmotic forces. So in certain environments, depending on the environment, the cell wall or the cell membrane, sorry, could expand, and the cell wall keeps the cell membrane from expanding to the point where preventing the cell from lysing. This is the image of peptidoglycan in our book. So this would be what you could look at in our book. NAM and NAG in blue and yellow, alternating subunits, and then cross-linked together. NAM and NAG have long names. I don't require you to memorize their names, but N-acetylglucosamine is NAG and N-acetylmuramic acid is NAM. Here we have the structure of glucose, and you can see here that the cell structure, or the peptidoglycan NAM and NAG, is derived from a glucose molecule. Here's another image of gram-positive bacteria. Here we have the thick layers of peptidoglycan. Here we have the lipoteichoic acid anchoring it to the cell membrane. And then the teichoic acids that allow certain ions to pass through the cell wall and gain access into the cell. You should be able to look at images, such as images found in your course pack, and label the structures and identify very easily a gram-positive organism. Gram-positive organisms have this thick peptidoglycan and the cell membrane. As you'll see here in a moment, gram-negatives have actually 3 layers. This is the image found in our book again for gram-positive cell walls. Thick layers of peptidoglycan, have the lipoteichoic acids anchoring it to the cell membrane, and then the teichoic acid. So again, the cell wall is located outside of the cell membrane. Gram-negative cells, like I mentioned, have 3 layers. They have the inner cytoplasmic membrane, they have a thin layer of peptidoglycan, and then they have an outer membrane. Thin layer of peptidoglycan. Outer membrane is composed of lipopolysaccharide and phospholipids, as you'll see here in a moment. The outer membrane also contains what are called porins. Porins are highly selective transport proteins, so they only allow certain molecules into the cell. There's periplasmic space in between the peptidoglycan and the cell membrane, and periplasmic space between the peptidoglycan and the outer membrane. And gram-negatives appear pink following the gram stain procedure. Here's an image of gram-negative organism showing the porins in the outer membrane. Lipopolysaccharide is the outer leaflet of the outer membrane, and where we have phospholipids on the inside of that inner leaflet. Periplasmic space, thin layer of peptidoglycan, periplasmic space, cell membrane. This is the image of the book that I modified slightly, adding these little tubes here indicating the porin proteins. Outer leaflet lipopolysaccharide, inner phospholipids, thin layer peptidoglycan, and the cell membrane. Okay, again, the outer membrane is outside of the peptidoglycan, and the peptidoglycan is outside of the cell membrane. Here's an image showing you side-by-side comparison of gram-positive and gram-negative. I believe one of these is found in your course pack. So right here, by looking at these two images side by side, see the gram-positive here on the left, thick layer of peptidoglycan, and then the gram-negative, thin layer peptidoglycan. But again, you should be able to recognize the gram-negative very easily by the indication of 3 layers: outer membrane, peptidoglycan, cytoplasmic membrane. Gram-positive, cytoplasmic membrane, and then thick layers of peptidoglycan. So what difference does it make if a bacteria is gram-positive or gram-negative? The main reason that it's important for people going into healthcare is it aids in the choice of antibiotic used for treatment. Some antibiotics will target the peptidoglycan of the cell. Antibiotics such as penicillin disrupt the formation of peptidoglycan. So gram-positive bacteria are more susceptible to penicillin derived antibiotics, whereas gram-negatives have the outer membrane that serves as an extra layer of protection. So the outer membrane prevents penicillin from actually coming into contact with the peptidoglycan. Not all bacteria have cell walls. This image shows Mycoplasma bacteria, and Mycoplasma are referred to as what is called pleomorphic in their cell structure or morphology, indicating that they have varied shapes depending on their environment. Some bacteria produce what's called a glycocalyx or a capsule. A glycocalyx is a polysaccharide secretion or a mucus layer that surrounds the cell. So this would be outside of the gram-negative outer membrane or gram-positive cell wall. Again, not all bacteria produce a capsule. The function of the capsule is to protect the bacteria from phagocytosis. The capsule material, the polysaccharide, is made from materials found similar in the human body. So phagocytic cells actually don't recognize the bacteria as a foreign object. Bacteria, or the capsule can help bacteria adhere to surfaces. So plaque on teeth, often those bacteria are able to form a capsule that helps them attach to the teeth, and then actually those bacteria produce what's called a biofilm that further allow attachment. And also protects the cells from desiccation or drying out. Some bacteria have flagella, which is used for motility, and these are too thin to be seen with a light microscope, so we have to use a special stain which will actually attach to those flagella proteins in order to highlight those using the light microscope. The flagella can come in a variety of arrangements. So here's Paratrichous flagella, indicating that they're found all over the bacteria. Polar, just at one end. They can be tufted, or they can be at both ends. There are also Pili or Fimbriae on some bacteria. Again, some bacteria produce flagella, some do not. Some bacteria produce Fimbriae or Pili, and some do not. They're shorter and more numerous than flagella, as you can see here in this scanning electron microscope image. Here you see the long flagella, and then short, numerous pili, aiding in attachment to surfaces. Some bacteria produce specialized pili, known as what's called the sex pilus, and these can transfer genetic material such as plasmids through a specialized pili into another bacteria. We call them sex pilus, but it is not a form of sexual reproduction. Here they indicate this pili here highlighted as a conjugation pilus. This would be what we refer to as the sex pilus that I just mentioned. So here, small pieces of DNA, such as a plasmid, can transfer from one bacteria to another. So if this bacteria over here on the left is an antibiotic-resistant bacteria, and that gene is found on the plasmid, it can transfer that plasmid through the tube into this other bacteria. That new bacteria has the plasmid, and it could now be antibiotic resistant. A couple of special cell structures would be endospores and biofilms. Endospores, very important. You will find questions on the short answer portion of the exam about endospores. And an endospore, in general, is a resistant dormant structure formed by some gram-positive bacteria. So this is important. Only some gram-positive bacteria can produce these. Organisms from the genus Bacillus and Clostridium are clinically significant genre of bacteria because they are disease-causing bacteria. So Bacillus anthracis, for example, Clostridium tetani, Clostridium botulinum, Clostridium difficile are all endospore-forming bacteria. The cells that produce endospores do not have the spore at all times. The cell exists as what's referred to as a vegetative cell, which is an actively growing cell. A lot of people confuse vegetative cell with vegetative state. That is different. So vegetative cell is an actively growing cell, whereas a vegetative state, which somebody might be in as a result of a car accident, for example, It would be somebody that is not able to take care of themselves, so they're not active. And what happens is a vegetative cell undergoes this process of sporulation, which is about an 8-hour process. So the cell does this when conditions are less than favorable. So, in the response to nutrient limitation, high cell density, or potential other environmental factors can trigger this process of sporulation and cause the vegetative cell to form an endospore. Once they are in the endospore state, there is no detectable metabolism, meaning that they can survive for long periods of time as the endospore. The thing about endospores is that they can germinate and develop into a vegetative cell again, um, which makes them clinically significant because they are resistant to certain environmental factors such as uh, chemicals, disinfectants. They can stay on surfaces for a long period of time, and then once they come into contact with a favorable environment, such as the human body, they can germinate and cause disease. So we have what's referred to as an endospore cycle. We have a vegetative cell undergoing normal activity uh, in an environment. There is limited nutrients, so those vegetative cells are basically fighting for access to nutrients. Once those nutrients become limited, they undergo this process of sporulation, 8-hour process, and after sporulation, they are that resistant dormant structure, the endospore. The endospore comes into contact with favorable conditions, such as the human body, like I mentioned, and they can germinate and become a vegetative cell again. The endospores can survive for long periods of time, years or even centuries. So there's evidence of endospores being germinated from millions of years ago. And they're resistant to extreme environmental conditions such as heat. So we actually have to use what's called an autoclave to destroy them. So an autoclave will heat the samples up to 121 degrees Celsius with 15 pounds per square inch of pressure. For about 20 minutes will deactivate the endospore. They're resistant to chemicals such as common disinfectants, desiccation, which is low moisture, UV radiation. The thing that people think, though, is when they see resistance in this class, is they think antibiotic resistance. Antibiotics target vegetative cells. Endospores are not treated, so to speak, with antibiotics. So it's the vegetative cells that once they germinate and cause disease, then you would have antibiotics being used to treat the disease, such as anthrax. Biofilms would be the last special structure, and this is a community of organisms. They can be a variety of microbes, both prokaryotic and often eukaryotic cells within this biofilm, which allows them to attach to solid surfaces and grow in masses. They can grow on rocks and pipes, teeth, medical implants, and cause disease. Here's an image of a Staphylococcus aureus biofilm on a catheter. So here you see this, the biofilm itself is this polysaccharide secretion, very thick layers of this mucus-like material, allowing the bacteria to attach to the surface of the catheter and actually have little tunnels which help transport nutrients to the cells themselves. This concludes the Chapter 3 lecture video. Like I said, this video is this chapter is very important for the entire semester. Make sure you use the study guide to learn the structures and know the functions of what we just discussed.